Any other sightings to share? All right. Well, as some of you have already done, we ask that for our speaker that you mute yourself. And if you're invited to ask a question, you can unmute yourself to do that, or you can use the chat function and throw a question my way, because Margie might not see it. And so then I can, to stop things, interrupt kindly, gently, hopefully, in order to present questions. So thank you all for being here, and thank you, Margie Menzies, for joining us. You may know her from just being up at Hodge and seeing her there, or for various education opportunities and field trips as well. And so we're very happy to receive an update about the good work that you do at Hawk Ridge. And thank you.
Awesome. Well, I'm going to share my screen here. We did one test run. That should do it. There we go. And get the slideshow started. There. Everybody seeing a Duluth Audubon headliner there? Hawk Ridge Counting for Conservation? All right, yeah.
We see it.
Awesome. All right. Well, I'm excited to be here. I told Jeff, I'm like, hey, I get to talk to the hometown crowd. I don't get to do that very often. I suppose to mostly speak to out of towners, but there's a hometown crowd in there too, but they're in smaller numbers. So anyway, it's kind of fun to be here with all of you. I love talking birds, so no problem there. I thought what we'd do tonight is just have a quick some of the highlights from last fall, updates, some things that happened. Winter wonders, we've done some owl banding over the winter, and then spring migration is already underway, so I can give you a brief update on what's been happening with that. And I thought that you would enjoy hearing about some of the research efforts that are happening at Hawk Ridge. There is some really way cool, exciting stuff happening. So I thought that was kind of the run of things that I would do for this evening, and definitely willing to hang around for some questions and discussion afterwards too, if people are interested. So yeah, Hawk Ridge, we're here in Duluth in this amazing place. And if there's a place to have a bird observatory, I can't think of a better one. And yeah, you're all familiar with our mission to protect all of the birds that migrate in the Western Lake Superior region through research, education, and stewardship. Our last fall was a little different in the COVID world. I was very pleased with the big full-size bald eagle things that we put our COVID cautions on, and we'll have those for a variety of educational things now. But we did do things slightly different, tried to encourage people to stay socially distant from each other and wear masks and that sort of stuff. So looked a little different this fall. We didn't do much for public programming, a little bit, but did still do bird demonstrations and things like that, but the public weren't able to release them this year, which is kind of a bummer, but I have to be kind of happy about that because it allowed me to have lots more birds in my hand, and I released a lot more birds than I have in a long time over the fall. So there were little nippets of things that kind of made it really nice. And ordinarily I spend a lot of time on the computer doing scheduling and working with people on programming, and I mostly spent every day in the fall outside looking at birds and talking to people all day. Not a tough deal. It actually was very nice. And so we were able to successfully make it through the season with really not much differently than usual as far as the counting and banding. Education was the only thing that was really significantly impacted. We did banding as usual last year. I'm holding one of the highlights. That's a red-shouldered hawk. That was the 5th ever caught at Hawk Ridge, and I got to release it and do a little showing off with folks. We had a number of species this year that we banded more than normal. It was a good year for northern goshawks. it was an exceptional year for rough-legged hawks. We'll talk a little bit more about that in a bit. We had that unusual red-shouldered hawk, and our second ever golden eagle was caught by Abby Valine in our Moose Valley station. So we were very, very excited about that. Take a look at that big one. And if you want to get an idea of those talons, look at Abby's little hand there holding those talons and imagine being on the receiving end of something like that. Very exciting to catch. Like I say, it was the second one ever. So that was pretty cool. Definitely a highlight. We had a great owl banding season, and I was laughing as I was looking at this. Every single one of the owl species that we banded this year was more than normal. We had an exceptionally good northern saw-whet year. We typically band about 1,200. This year it was over 1,600. About double the normal number of long-eared owls. 179 this year, a new season record. 18 barred owls, 1 great gray, which is highly unusual for at the station. We usually do that over the winter, but not at the station. 6 great horned owls, and our little eastern screech owl. Now, those of you that know about screech owls in Minnesota know that screech owls are generally not in northern Minnesota. So this picture of this little one in the lower right-hand corner is a bird of some infamy at Hawk Ridge. It was banded in 2018 because the banders at night kept hearing it. And so they actually were able to call it in and caught it, banded it, and the bird was back again in 2019. And yes, indeed, back for the 3rd time in 2020. So we keep hoping that it coaxes a mate up that direction and we start getting more than one eastern screech owl. But for right now, this one, I think it's a pretty safe bet to call this one a resident because we've caught it now 3 years running. So fascinating story. So really, really cool kinds of stuff going on in the banding as always. Count wise, it was an interesting year for count things. Some things were better than average. I put those on the left side. We had a new single day high for turkey vultures on 854. Ordinarily, we get about 1,500 turkey vultures. We had more than 2,300 this year. And rough-legged hawks, oh my goodness, did we have them. A new season high for those at 1,948. The old record was a little over 1,100. And a new single day high of 583 on a single day in October. Note that that's higher than the season average, the 10-year season average. So yeah, it was an exceptional year for rough-legged hawks. As I mentioned earlier, a good year for goshawks. Broadwings were a good year. We were about 5,000 and some over normal for broadwing hawks as well. Golden eagles. The interesting thing, if you look at the less than average side of the spectrum, some of these birds are less than half of what we ordinarily see, which is an interesting thing. And the other interesting thing for me when I look at that is that nearly all of those birds are early season migrators. So I don't know if there was something going on early season that shifted some of those birds a different direction. Red-tailed hawks, I did a little snooping on that one. That is a later season migrator, and ordinarily we see close to 7,000 in a year, and we didn't come anywhere close to that this year. And so I did a little snooping on Humana, the Humana website, and looking for other hawk count stations in our neighborhood to the east or the west. And it turns out that, yeah, in looking at that, Mackinac Straits to the east of us had just a truly exceptional red-tailed hawk year, like 3 or 4 times what they ordinarily have, although their count has only been in full swing for 4 years. And so that's a little bit, you know, we need more data to say that in certainty, but I think a lot of birds went east this year for whatever reason. So interesting stuff going on there. Always interesting. The total count, I was messing around with that stuff the other day when I was putting things together for this show, and I was like, okay, well, I have a whole bunch of birds that were less than average, and I have a whole bunch of birds that were more than average. Well, the more than average numbers more than made up for the less than average. So yeah, we had a slightly higher than average, the 10-year average of 61,150 birds. We had 62,700 and some. So yeah, interesting stuff. It's always interesting to look at those count numbers and think about differences from year to year. Moving on into the winter, we had a better than average year for great gray owl sightings in the Northwoods here, but it was not an eruption year. Over the winter, our banders did successfully band 9 great grays. It was a lousy year for northern hawk owls. We can usually count on several, at least a couple to several in Sax-Zim, and there was only 1 this year. They did band it. But yeah, that was the one and the only. There were rumors that it was carrying either a really, 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 really big territory, or there were 2, we just could never see them successfully enough to say, okay, there's one that's keeping this territory up towards the greenhouse and another one that's keeping it more towards the bend on Highway 7. So yeah, we're not real sure yet what happened with the northern hawk owls, except that they weren't here. There have been some very recent late season sightings of boreal owls, but not very many. Northern saw-whets are starting to be sighted more regularly these days. So we have seen a few more of those. But those great grays, they're just plain big. When you have one of those in your hands, and this picture on the left is the first time I held a great gray in my hand. I was a very happy camper. I'm not so sure about the bird, but I always like to show this photo because it very clearly shows that the height of the bird goes from roughly my waist all the way up to my forehead. So that's 2+ feet. And yeah, just holding something that large in your hand is pretty amazing. And yeah, they have a pretty good set of legs on them as well. Not quite as big as those golden eagle ones, but it still takes a fair sized band to band these great grays. We of course do all of the normal data recording that we would do at the station. We just are doing it roadside kinds of things when we're doing the winter banding. Here's a northern hawk owl for you that's got caught and was banded. This was not from this year. This was from a previous year, the year before, but still amazing birds. And we really enjoy the opportunity to try and do that. I'll tell you more about hawk owls here in a bit because there's some research starting with them. So yeah, believe it or not, spring has sprung, and we are off and running with the spring count. I threw this slide of our website just in case you don't know where to check things. You can go in and you can look at what's happening with the spring count in 2 different ways. You can get the live by the bird happenings on the live count, so you can click there, or you can look at the more raptor centered day-to-day, month-to-month count here. So they both have their pluses and minuses, and we'll show you a bit more about that in a second here. Our spring count doesn't happen in the same location as the fall count, partly because they don't plow the East Skyline Parkway spot, and it doesn't open up as a road until mid to late May. So it's much easier to count on the West Skyline, and the sightings are better for the birds coming from the south. So we have two locations that we use. Depends on what's happening. I always say the answer, my friend, is blowing in the wind. If we are getting a good northeasterly wind or a wind from the east, both of them very chilly coming straight off the lake, we're going to be counting from Thompson Hill. Yeah, you better have your layers because it's usually pretty chilly when the wind is coming right straight off the lake. But it's a better sighting spot for a lot of these birds as they're coming through. They're taking advantage. Spring migration is different than fall migration in a lot of ways. And one of those ways is those birds are headed for breeding grounds and they're in a hurry. They want to get there before somebody else takes over their territory if they're a returning bird. So they're much more about business in the spring. Get there or die trying in some ways. And that flying regardless of the wind, which is something we don't see very much in the fall, but we do in the spring. They fly regardless of the wind because they're really trying to get where they need to go. And they can make use of the cliff faces because of natural winds that travel uphill at those locations. They can get a little natural lift off of those ridges. And that's why the Thompson Hill site is such a good spot. We do get natural lift off the ridges at the other location as well, which is Rice's Point on West Skyline, just below Inger Tower Park. But we're there more often than not with all the other wind directions. So it's just when it's blowing off the lake that we're at Thompson Hill. So yeah, it looks about like this year. This is not this year's picture, but yeah, this year I think our snow is going to disappear pretty quickly, although we did get a new influx last night and I haven't been out there to the count location since it snowed. So it does look a little different, but it doesn't look like it did in 2019. This is the 31st of March, 2019. Yeah, it could look like that again, but we had just a massive influx of snow that winter, and it took a long time for the ice and snow to clear out. You can see that the lake is still largely frozen over on this inland side or close to Duluth end. So here's a couple of up-to-date things thus far this season. 741 bald eagles, and 24 golden eagles. So the eagles are coming through, and then a random scattering of some of the others. Cooper's hawk, 2 of those so far, and a northern goshawk. Some red tails are beginning to work through, and some rough-legged hawks. So yeah, things are off and running. This is the Humana side. So this is what that will look like if you pull that side. If you pull up the live count, you can look at this graph, which will tell you all of the species that we've seen so far. I don't expect that you're going to read all those numbers, but we do count all the birds that we see going by. So this will include all of the other birds in addition to the raptors. So that's happening.
Margie.
Yeah.
Somebody who can't stay for question and answer kind of time has a question just about what do you make of some of these good years for some of these high numbers that we've been having? And what do you make of the fact that the red tails were found, much further east than were expected?
Yeah, well, one of the things that I tell people all the time is that 1 year's record is not enough to make really conclusive assumptions about what happened. Yeah, at Hawk Ridge, yes, we are in our 50th year of collecting count and banding data. And you can look at 50 years and make some conclusions about things. But individual years is hard. Why the red tails went east more than likely was a weather thing. And the winds were just the wrong way. And a whole bunch of them went the other direction because the winds were the wrong direction. Honestly, though, this year was good wind predominantly. I mean, the last couple of years prior to this last one, we've had a lot of southwesterly winds that tends to slow everything way down and make the birds go around. And we really didn't have as much of that this past fall. So I can't give a good explanation for why, especially those early season birds, because we had just a phenomenal month of September. Things did cool down quickly in early to mid-October, but then it rebounded towards the end. So yeah, the red tails I'm not so sure about. Rough-legged hawks, I can say with some authority, at least I think on that one, that they must have had a really, really successful breeding year. But again, we can't be absolutely certain of that until we spend a lot of time looking at everybody who counts and looking at all of the data. And is everybody seeing more than normal rough-legged hawks? And I haven't had time to look into that, but you can on that Humana site. When you go on the Hawk Ridge spot, it'll pull you in and you can look at that. Let's see, let's go back. Up above this, there's a pull down menu and you can change your location. So you can pull up Mackinac Straits or you can pull up Whitefish Point and you can see what they're seeing and you can compare and contrast. I always say that there are many, many, many master's degrees waiting to be written from looking just at the data.
Apparently you're blind. I didn't realize that, but you are. You're never interested in looking at anything.
So I think we're having some people that are not muted, I think.
Oh,
is
okay.
the issue here. So
all right, okay.
we'll work on that.
All right, we will keep
we
going. I thought I would stick a little spring migration quiz kind of question in here. All right, there are 4 bird pictures here and there's John doing some counting there. This is a red-tailed hawk. This is a northern harrier. This is an osprey, and these are tundra swans. And your job is to, in your head, put them in order. Who's going to come through first, second, third, and fourth? And I'll give you just a couple of seconds here to kind of organize that in your head, and then we can talk about that. It is kind of a parade of birds in the spring.
May we venture a guess?
Sure.
Swans, red tails, no, swans, red tails, the harrier, and then the osprey.
Sounds like a well-schooled individual there. That's what I would say too, is the swans are going to be first, and then on to the red tails, and harriers and osprey. Well done. In fact, here's a little chart that I'm working on. We've just finished making this up, and we'll eventually get it on the website so it's accessible. But it's interesting, when you look at birds in the spring, more often than not, the adults are going to come before the young birds. So on this graph, the adults are the green bar and the juveniles are the blue bars. And you will see that in most cases, the juveniles come later. Now, osprey, that was one of our birds. Why don't we have any juvenile osprey coming? Well, the truth of the matter is that osprey are pretty darn smart. The young ones tend to stay south and they don't migrate north until they're ready to breed. And so the juveniles don't come north, which is an interesting thing. Kestrels, what's on with that one? Well, turns out that we can't tell a difference when they're flying by between the adults and the juveniles by this time of the year. And so can't call them. So that's why there's no blue bar there. But for most of these birds, the adults will come before the juveniles. And that's kind of the reverse of what happens in the fall. In the fall, we see the juveniles before we see the adults. So another way that spring is different.
Thank you for
So
the little tidbit on ospreys. I wonder how many of in our group here tonight knew that. I didn't know that, I'll admit. And so thank you for that little tidbit.
oh, sure. Yeah, hang out with a bunch of bird nerds and everybody's likely to learn something from somebody. So yeah, I always love the bald eagles. They are the ones that get the party going for sure up here. Mid to late February is when you get started heading north if you're a bald eagle. Our count officially begins on March the 1st, but Frank usually can't help himself and starts a few days early in February, particularly if the birds are moving, and that is no different this year. He did start in late February. We see more bald eagles in the spring count than we do in the fall. You can compare the numbers there by about 600 birds or so, 650 birds. We set a new spring count record of 7,727 in 2019, and also the most in a single day in 2019 of 1,076. And I was out there that day and it's just phenomenal. Just imagine yourself looking at that whole industrial park skyline and okay, there's a kettle and it's all bald eagles over there. There's another one, there's another one. And scanning your binocs across the sky that day, you could easily count more than 100 bald eagles in just a scan if you're working hard at it. So just a phenomenal number of bald eagles came through on that day. Last year we had a less than average day, but this year is off to a good start. I looked at previous season numbers and our numbers at this point this year, and we're a little ahead of ourselves. So it's a good thing. So far, so good. But the peak flight for the bald eagles typically is mid to late March. So there is lots more to come. And we will see bald eagles actually all the way through the spring, almost all the way to June. And there's other things involved with that. Some of the southern birds that breed at points way south actually come north when they're done breeding. I think probably because the fishing's better and it's cooler. And yeah, I wouldn't hang in 100-degree high humidity weather if I had a choice. So yeah, I always call them the tourist birds, but they'll start arriving even in late May into June. Turkey vultures are another bird that we see more in the spring than we do in the fall. So always fun to see those guys. And another really interesting thing that I had brought home to me really, really dramatically, and I have seen it a number of times since. Bald eagles and turkey vultures, as far as wingspans, are pretty much equal. Maybe the bald eagles have got an inch or two on the turkey vultures, but not by much. But the turkey vultures weigh less than half, sometimes a third as much as those bald eagles. So it's not unusual on a spring day when they're both coming through to see, with a light wind, the turkey vultures just blithely bopping along, soaring like crazy, and the bald eagles are all pumping like crazy just to be able to fly at all. So it's a real interesting thing to see, that difference in weight and what it does in their flight patterns. Other spring faves, sandhill cranes, they've been seen along the St. Croix River in the Twin Cities area, so it won't be long before we see them up here. Peregrine falcons, that's a bird that we typically see less of in the spring than we do in the fall, but we have resident peregrines that live in the harbor area, and so our counters have to be really cognizant of who's a resident and who's migrating by watching their flight patterns. And one of my favorites, the pelicans. As the St. Louis River thaws in the spring, we will see lots of these guys, and some of them will hang for a week or two. That picture in the upper left-hand corner is from Chambers Grove area last spring. It's not unusual for the pelicans to hang down in that neck of the woods for a couple of weeks while they're waiting for the inland lakes that they're headed to, either at points in Canada or inland from us here and more over towards the Grand Rapids area and north. There's some breeding white pelicans in those lakes. So they're waiting for those lakes to thaw out. And finally, yeah, we get all of our warblers and shorebirds and things like that as they travel along. And with that, I'm going to slide into our research efforts. And like I say, there's a lot of cool things going on. MAPS is one of the research projects that we have going. MAPS stands for Monitoring Avian Productivity and Survivorship. As a passerine bander, this is my fun time of the year. This is a breeding season program, and we get to know these birds literally a little better than we do all of the migrants that come through. Here's some of our normal fare in the summertime. In MAPS, what you do is you set the nets in the same locations, and there are 10 sampling sessions during the breeding season, or sorry, 7 every 10 days. I got my numbers backwards there. Seven sampling sessions once every 10 days. And yeah, over the years, you'll have birds that come back to the same nesting location year after year. So we can keep track of those. And we can also, by an analysis of the birds that we catch later in the season, we can analyze and assess how successful a breeding season it was and how many young they're likely to have had, or at least begin to work with some of those things. So yeah, some of our normal fare, flycatchers, we always have alder flycatchers. Golden winged warblers, we always hear them and catch them early in the season, but then they disappear and they don't stay through the whole breeding season. We haven't convinced them that they should stay, but I think one of these years they will. Black and whites are normal, and lots of hummingbirds and indigo buntings and cedar waxwings, all common stuff that we band a lot of in the summertime. Summer 2020, we were able to successfully continue our MAPS program with some new station protocols, and we're able to band 269 new birds and 85 recaptures, some from the same season, but many from previous years. And that's the really exciting thing. Look at this red-eyed vireo, and no, it goes, most of them go all the way to South America and come back to the exact same location. That just blows your mind. Yeah, so that's one of the research projects. I stuck in these pictures of that John took last winter of a merlin getting the better of a pigeon. I put this one in because, yeah, it's all in the moves and in the feathers. For a number of years, we've been working on monitoring the amount of mercury present in the feathers of raptors. That was a research project started by Ted Kale, and has been continued. And we've found, interestingly enough, that it doesn't go all the way across the full spectrum of raptors. Turns out the birds that have the highest levels of mercury are the birds that eat other birds, like merlins, and another that I'll mention here in a little bit. So yeah, interesting to do that. So we've been doing that with feathers in raptors. And as we analyzed that, we said, hey, you know, we should back it up the food chain and see what we're finding at the passerin level. So we've had a list of passerins when we're banding during migratory season that we collect feathers of in addition to the raptors. Birds like rusty blackbirds, we were talking about them earlier. This is a northern water thrush and a western palm warbler. Of the birds whose feathers we've analyzed so far, those are some of the birds that have had the highest levels of mercury. So we're just getting started on this. This is a fraction of the feathers that we've pulled on the passerin level, but what we're finding is that some of these smaller birds do have fairly significant levels of mercury. On this graph, the line is kind of the average, and then these are outliers, things that were way out there. So here in this northern water thrush, you can see that this is about average, but we had some that had significantly more. So it's all part of the story that we're trying to figure out, following back the food chain. And here's another one for you. These are sharp-shinned hawks, and sharp-shins are also bird consumers. And we've found from the mercury levels that they do also have significantly higher levels of mercury in their feathers. And this is Lula, one of our great volunteers. She and her family come out frequently and help us out. And I put that in there for perspective. And you're probably all wondering why in the world I stuck a turkey picture in there. Well, let's move on to another research effort, and I'll tell you why.
Of the 287 cloaca swabs I analyzed,
I did not know that was going to be loud.
At least one prey species.
Can you hear that?
Swabs with prey DNA detected one prey OTU. 8 swabs detected 2
Yes.
prey OTUs.
Oh goodness.
Of the 11 species,
I don't know how to shut it up.
The next interesting case was a sub-obstruction hawk swab.
Seriously. Okay, well,
these 3 samples were all collected in the
that was a highly
Now, if you
unintended
aren't familiar with
consequence.
hawks and you're wondering if one could kill an adult turkey, the answer is no. So what's the deal here?
Let's see. I'm thinking, yeah, I asked permission to, these are from videos, and if you want to watch the full videos from some of these researchers, MOU has their December paper session, and several of these researchers presented in that, and I asked their permission to filch things from their slideshow, but I didn't realize I was getting sound as well. That's probably good, but I don't want to take that much time with it. So I apologize for that little technical glitch. It certainly didn't show that to me when I was working on this thing. But anyway, one of the things that Lisa Brulette has been doing with her master's program is studying what these migrating birds are eating. We've done a lot of work on raptors and what they eat in their breeding territory, but there hasn't been a whole lot of work done on breeding or on migrating birds and what they're eating. And so she hypothesized that she could take swabs of the cloaca. Cloaca is the outbound circuit for the bird, the cloaca. And if we were able to take swabs of birds that we captured at the banding station, swab the cloaca, and then be able to do DNA barcoding processing to figure out what they've been eating. This is a really interesting thing. There is a database of prey DNA out there. Lisa found that She couldn't match perfectly every single one of them, but she could for some. And so she learned quite a lot about what these birds are eating. She did learn that it was a no guarantee that if you swabbed a bird that you were actually going to find prey DNA. Only 18.4% of her diurnal raptors yielded anything, but the ones that did told her she found some that 43 that had one type of prey, at least 2 that had 3 or more types of prey in their squab, and 8 that had 2. So interesting stuff. So if you look at these birds, sharp-shinned hawks, blue for birds, we knew that. So yeah, some interesting things there. Coopers are kind of a 50-50 mix, as are northern goshawks. Red-tailed hawks are more on the mammal side of the spectrum, but they do take an occasional bird. And northern saw-whets are mammal types. So yeah, she was able to connect things pretty directly.
The next
On to the
interesting
turkey
case was a
story.
several sharp-shinned hawk swabs
So
that
I'm going to
contained
push
turkey
this
DNA.
forward.
These 3 samples were all collected in the spring.
And now I'm going to back up.
Familiar with
Oh,
sharp-shinned hawks,
sorry.
and you're wondering if one could kill an adult turkey.
Well,
The
we'll
answer
just
is
listen.
no. So what's the deal here? I thought of a few possible explanations. First, that they had simply eaten a turkey chick. But wild turkey chicks in Minnesota and Wisconsin don't hatch until early June, and these samples were collected in late April and early May. And free-range turkey farms in our area would not be letting young chicks out of the barn at that time of year because of the cold. Wild turkey chicks do start to hatch in late April in southern Texas and Florida, but it would take several days for a sharp-shinned hawk to fly from the southern US to Duluth. And while we don't know how long prey DNA can hang out in the cloaca for, the average gut retention time for raptors is about 6 to 8 hours. So it doesn't seem likely that a swab would detect prey eaten more than 24 hours before the sample was collected. The first case is of a northern goshawk swab that had opossum DNA. I thought this was really interesting because if you're from Duluth, you know that opossums are not very common in our area.
Okay.
First and foremost, a controlled study would be the best way to answer questions like how long prey DNA persists in the cloaca.
Okay. Hopefully this won't happen again. My apologies. But yeah, she did find turkey in sharp-shinned hawks, 3 of them. So not just once, 3 times. So yeah, the idea that hadn't been documented up to that point of sharp-shinned hawks feeding on carrion becomes a different story. Perhaps they do during migration. It sort of makes sense thinking about you're out of your normal territory. Hunting is going to be more difficult. Maybe opportunity at carrion is a good thing to take advantage. So yeah, perhaps we're finding that. And then how the northern goshawk got possum is another question. Another of the northern goshawks that she got had porcupine. You can put that one in your brain and rattle it around for a little while. But I think overall, probably the end result is that they are taking advantage of carrion. So yeah, time is flipping away on me here, so I'm going to keep moving. Here's another research project Emily Pavlovich is working on, another grad student from UMD. We see thousands and thousands of birds every year, but we would love to have more information about where they're coming from. And it turns out that her study is going to connect their breeding territory to what kind of routes and flights they take in migration. We know that birds accumulate things that from their environment. You are what you eat and or drink. And she's going to be studying stable isotopes in birds, predominantly hydrogen and water. There's an extra heavy version of water that occurs with deuterium, which is a hydrogen ion that has an extra doesn't ordinarily have a neutron in the nucleus of hydrogen, but in deuterium they do. And we've been doing deuterium studies on passerines for a long time, but nobody's really done it for raptors. So Emily's going to be working with it with raptors. We do know that you can make maps like this isoscape of where in different regions of the world, there's different levels of deuterium present in the water samples, and it builds up in the feathers accordingly. And over the years, with passerine studies, we've been able to make essentially a feather isoscape and have had samples of feathers from known locations. Now we're going to be able to take the raptor feathers and process them and compare them to this known database and see if we can figure out A, where were these birds bred, and B, what are they doing for their migratory patterns. So last fall, she was very successful in collecting feathers and has a lot of processing to be doing on all of those feathers, 438 sharp-shinned hawks and saw-whet owls, and a significant number of red-tailed hawks as well. So she'll be taking a look at the results from those and beginning to perhaps get some answers on where those birds are actually coming from. Pretty amazing stuff. Lots more questions can be answered in there. Here's another fascinating project underway. This one is Ali Pisano's project on red-tailed hawks. If you look at red-tailed hawks on the map, they're one of the most widely distributed birds in North America. We know that there are a number of subspecies, 7 in the contiguous US, and over the years, we have the Borealis subspecies, which is considered the eastern bird, and the Calurus, which is considered to be the western bird. But here's where it gets interesting. We know that there's also variance in the amount in the plumage of these birds, and there's a dark morph and light morph bird for Calurus, but not for Borealis, or at least that's what they say. But at Hawk Ridge, we know differently. Theoretically, we should be getting only Borealis birds coming, or predominantly Borealis birds. And we do get Borealis birds. We do see some Calurus birds too on occasion, but we see a significant number of dark morph birds. And if these are Borealis, what the heck? There should not be dark morph birds, or at least according to our current scientific knowledge, the Borealis does not have a dark morph. So where are all these dark morph birds coming from? Are they wandering Calurus birds? Are they dark morph Borealis that's been lurking in the background and nobody's ever documented before? Is it a hybrid? Or is it another option? We have a not official subspecies of Abeticola. And here's the story about Abeticola. We know that red-tailed hawks that breed up in the boreal forest with the spruce and fir are more darkly colored. You can see in the Abeticola slide here that there's a lot more color in the upper breast and inner feathers on the wings on the underside versus the typical Borealis, which is much whiter. We know from our data of banding and from counting that about 60% of the birds that come through Hawk Ridge in the fall are Abeticola. So this is what Allie's working on, taking measurements and photos, and we've taken blood samples for a number of birds, and we'll be analyzing the DNA. And she also is doing some satellite tracking. Last fall, she was successfully able to get a satellite tracker on one light morph, a beta cola, that spent the winter predominantly around the Rice Lake, Wisconsin region. And then later this winter, here maybe about a month ago, they were able to successfully catch a dark morph down closer to the Twin Cities and get a satellite transmitter on that one. She has 2 more transmitters that she wants to put on, one on another dark morph and another on a light morph, a beta cola, and get some idea where these birds are going for breeding. So way cool stuff. Northern hawk owls, another grad student's working on that. Hannah Tutongi is looking at an owl that hasn't been studied very much, and wanting to know why in the world do they come down here into northern Minnesota for the winter, looking at vegetation, looking at baseline metabolism, and also doing some satellite tracking with those birds. Last winter was when she was really hoping to get started with that. This winter was not a good one for hawk owls in Minnesota, so we're hoping she has better luck next season. And finally, one last study that we have going, we have found that there is some potential correlation between mercury in feathers and PFAS. Now, what is PFAS? Well, it's a large grouping of chemicals, have big long carbon chains, but the carbons, instead of having hydrogens attached to them like they often do, have fluorine molecules attached, which is a really, really, really strong bond. And if you get a big string of chlorine with carbon with fluorine attached, you have what they call a forever chemical, because it's very difficult to get those to break down. They're very common and they're also very persistent. They're in everything. Teflon cookware and textiles and waterproofing, firefighting foams, and all sorts of stuff. And they were thought to be harmless in the environment. But you may remember 3M settling with a number of communities in the Twin Cities about PFAS accumulating in the water table and in their drinking supply. Matt Ederson, who's our chair of our research committee, has been starting out, we've got an LCCMR grant to study this, and we've been taking blood samples from birds. It is a bioaccumulator in the system and some cause for concern. Blood work that's done separates the serum from the red blood cells, and the serum is what's actually tested for the PFAS. And they did 83 northern goshawks from this fall and 23 Sharpshin hawks. And initial results show that in northern goshawks, the females have less PFAS than the males, but the juveniles have more than the adults. Very interesting. Sharpshin hawks, on the other hand, another accipiter, females have more than the males, and there's no difference between the adults and the juveniles. So we're in the initial stages of trying to figure out some of this stuff. And there's going to be lots more work coming on with this. We've pretty much got enough samples of blood, but just need more time to be able to process and figure out the results. So keep your ears peeled. There's lots more stuff coming. So yeah, so much more to know about our feathered friends. We hope you've enjoyed this tonight, and hope you'll come out and take advantage of some of the spring migration that's going on. We love volunteers, so if it's something that you'd be interested in getting involved with, I know a number of you are already, but if there's others of you out there, we would love that. We are indeed now in our 50th year of counting and banding, but also lots of other research as well. So I'll leave you with a parting shot from last fall, just for fun. This is a rough-legged hawk from one of our many, many, many
Margie,
it
one
was-
question about the PFAS in the sharp-shinned hawks. Is that possible that they can excrete them over time?
That's one of the questions with the sharpshins. The compare contrast between the goshawks and the sharpshins, as far as really interesting. With the goshawks, they think perhaps the females are able to unload some of that PFAS in the laying egg process, which might explain why the young have such high levels, because they've gotten it directly from mom.
Okay, that makes sense.
But there's some, ask Matt. He told me that story, and I will not claim to be an expert on PFAS or any of these research projects. Like I say, I'm just excited to learn more about it and tell people what we're learning. If you've got technical questions, ask Matt, but he will tell you that he doesn't think much of that hypothesis of the female depositing it in eggs. He thinks there's probably another route that it's happening, but we haven't figured that out yet. So yeah, and as far as the sharpshins, they're sort of an enigma at the moment of why they're different than the goshawks.
From what I've read, you can't get rid of PFAS. And
Exactly.
that they're also building up in Lake Superior fish, especially in alewives. I mean, of all things. So yeah, and in smaller fish, which was sort of a surprise in one article that was in the paper recently.
Yeah, it's a chemical that's everywhere. And we're just beginning to do some of this work on, what are the things, but we know that it is a neurotoxin. It affects, there are neurological issues for both humans and birds, and probably many other things as well. And the thought initially with those things is that they were basically harmless, but I think we're finding out that that's not quite true. So there's going to be much.
They were warning that they were warning in the paper that you shouldn't eat more than one meal of smelt every month. Right.
Yeah, well, and that's a mercury accumulator as well. Mercury in fish is another issue, and like I say, there's some potential correlation between the mercury and the PFAS. And so, yeah, there's a lot more work to be done, and we're just beginning to scratch the surface.
We have a question regarding what the fall education at Hawk Ridge is going to look like, if there's been any thinking about virtual programming or in person yet.
Well, we are hoping, with the way things are looking right now, with the vaccine coming out very successfully, we're hoping that we might actually return to somewhat normal fall programming. Now, that said, we have learned some things through virtual programming, and there are some things that are likely to stay. We've started this Pick a Bird Brain program, and that one's likely to stay. It seems to be something that people enjoy doing, and maybe we will continue to do that. So yeah, I think it hopefully will be closer to normal again. That's the way we're planning at this point. But yeah, as we're planning as well as anybody can plan at this stage of the game. We'll figure it out as we go, like we have been for a year.
Thank you, Margie. It was wonderful.
Yeah, thanks so much for coming, everybody. Happy to handle some more questions or other discussion topics if you like.
Yes, any other questions? All right.
Well,
well, the work of Hawk Ridge Bird Observatory with counting and banding and research, and the work that you do is so important and appreciated. And we are grateful that you've been with us tonight to share with us more about it.
Thank you. Thank you for giving me the opportunity. Like I say, it's nice to talk to the hometown crowd.
Mhm.
Thanks, Margie.
Yeah, you're welcome. Good to see
Thank
you.
you.
Virtually anyway.
You too. Yeah. Take
That was
care.
good.
Yes,
That
thank
was
you.
good.
Presentation. Thank you.
All
Good to
right.
hear about all that research.
Yeah.
Yeah. Yeah, lots of cool stuff happening.
Great.
Well, good night. Good night, everybody. Stay safe and sane out there.
Thanks.
All right.
You too. See you next month. Bye.
Bye-bye.
I got to figure out how to stop recording now. There we go.
Stop.